Good morning, everyone. Today we are going to learn multiple alleles. Multiple means two or more than. Alleles means it's a trait or character. A set of three or more alleles of the same gene on the homologous chromosomes to control a particular trait is called multiple alleles. So alleles are the pair of genes occupying a specific spot called locus on a chromosomes. So human have a 23 pair of chromosomes. Typically, there are only two alleles for a gene is diploid organisms. When there is a gene exists in more than two allelic forms, this condition is referred as a multiple allelisms. Allelisms refer to any of the several forms of a gene. These genetic variations arise usually through mutation and therefore are responsible for heredity variations. So they occupying a particular locus on the homologous chromosomes. Each of these alleles is a repetition of the original sequences with a slight changes. For example, a gene of synaptic meiosis in 8th chromosome of tomato exists in the form of 5 alleles as 1 or as 2 or as 3 or 4 and 5. A set of multiple alleles may contains 3, 4 or more membranes, all the alleles occupying the same locus in the homologous chromosomes. Since all these alleles are formed by mutation of one genes, out of several allelic forms of gene, a given diploid individual possesses any two alleles of the allelic series and its gamete carries only one alleles. The ABO blood group is controlled by multiple alleles. The following are the examples for multiple alleles. One is an ABO blood grouping. One is a ABO blood grouping. The second one is a RH blood grouping. And third one is a nature of wing in Drosophila. Then fourth one coat color in rabbit. Then last one self-sterility in tobacco. So first we are going to learn ABO blood group. Carl Landsteiner, an Australian scientist, discovered the ABO blood group system in the year 1900. In his experiment, he mixed different blood types and noted that the plasma from certain blood type produced agglutination or formed clusters which were caused by the absence of molecule on red blood cells and resulting in antigen to defeat that molecules. He then made a note the aggregation and divided the four types of the blood groups. For the discovery of ABO blood group, he was awarded the Nobel Prizes. So Landsteiner found a four type of humaning depend on presence and absence of antigen in the blood. They are A, B, AB and O. A group persons contains antigen A on the RBC. B group persons contains antigen B on the RBC. AB group persons contains both antigen A and B on the RBC. O group persons contains no antigen as the RBC. A group of persons as antigen A and a antibody B. B group of persons has antigen B and a antibody A. AB group persons as antigen A and B and no antibody. O group persons as antibody A. O group persons has both antibody A and antibody B. So in this clear that antigen A cannot coexist with antibody A in any man. Similarly, antigen B cannot coexist with antibody B. The synthesis of antigen A is controlled by dominant allele represented by the letter L. It means Lance trainer. Is used to harness to discover blood group Lance trainers. Antigen B synthesis is controlled by another dominant allele represented by Lance trainer LB. The absence of antigen is due to presence of recessive alleles represented by LO. The three alleles are responsible for inheritance of ABO blood group. They are antigen A, antigen B, and antigen O. So LO is recessive alleles. It is a recessive both antigen A and antigen B. So antigen A and antigen B are dominant alleles. They are co-dominant. In co-dominance, both genes expressions their character. None is masked. When antigen A and antigen B occur together in the same man, antigen A produced antigen A and B produced antigen B. So it occurs same locus, they called multiple alleles. So multiple alleles of a gene occupying the same locus on the chromosomes. Multiple alleles are the alternative form of same gene, so the influence of the same trait. A wild type of alleles is mostly dominant over the mutant alleles. While this type is considered the standard and all the alleles are considered variants. An individual can have only a type of alleles, homozygous, or two different types, heterozygous. A series of mutant alleles can exist in populations. They are generated by spontaneous mutations. So multiple alleles lead a formation of variant phenotypic trait for a character in the populations. So next we are going to learn applications of ABO blood grouping. So first we are going to learn blood transfusions. The transfer of blood from one person to another is called blood transfusions. Blood transfusion is a life-saving process in accident and operation cases. Loss of blood from the body is component by transfusions. Transfusions needs for persons met with an accident or persons undergoing a major surgery or persons affected by anemia. Successfully blood transfusions need a through blood group testings. The persons donating blood is called a donor. The persons receiving blood is called a receptor. O group persons is the universal donor. AB group persons is called universal receptor. So next we are going to learn a disputed parentage. So the parentage of disputed baby can be confirmed by testing the blood grouping of the double persons. Similarly, the claim of two mothers for a single child, as happening in the court of King Solomon, can be settled by blood grouping testings. Then third one is the identification of culprits. So in murder cases, the culprit can be confirmed if the culprit's blood strain is available at the places of murder. So transfer blood from one person to another person A serious loss of blood is termed in transfusions. In transfusions in the blood of donor and receptor are not compatible, coagulation and hemolysis of the donor corpuscles will taken places. While testing the compatibility with the reactions between the antigen of the donor's RBC and antibody of the receptor plasma alone are they taken into considerations. So next we are going to learn RH factors. So RH blood group was discovered by Landsteiner and Weiner in the year 1940. It is controlled by multiple alleles located in the same locus of homologous chromosomes. There are two groups of human beings, namely Rh positive and Rh negative. Rh positive persons contains an antigen called Rh antigen, a presence on the surface of RBCs. Rh antigen is rhesus antigen as it was discovered by in rhesus monkey. A rhesus antigen is also called Rh vector. The Rh negative persons does not contain Rh antigen. The Rh antigen has no natural antibody. However, Rh antigen can be produced artificially. An Rh minus persons, Rh negative persons develops Rh antibody when he receives blood from as Rh positive persons. Even a small amount of Rh positive blood, as small as 0.0 ml, can evoke the protection of Rh antibody in the Rh negative persons. The antibody, once formed, remains in the blood throughout the world. In the European countries, 85% of human beings are Rh positive and the remaining 50% Rh negative. In our country, 93% of peoples they have Rh positive and 7% of persons only Rh negative. In China, 99% of peoples have Rh positive and only 0.5% are Rh minus. There are several varieties of Rh antigen and antibody. The commonest Rh antigen is called antigen D and its antibody is called anti-D. So the protection of antigen is controlled by multiple alleles. The antigen D is produced by a dominant gene represented by capital R. When this gene is recessive, it cannot produce antigen. In the Rh positive persons may be homologous dominant, capital R or heterozygous capital R small r. The Rh persons are always homozygous recessive. Therefore, compatibility between donor and individual is crucial in this case as well as. So next we are going to learn. There are two views regarding the genes of Rh blood group. They are, one is the Weiner's theory, another one is the Fisher's theory. So Weiner's theory means is nothing but Weiner's proposed that Rh blood group is controlled by 8 multiple alleles, namely a small r, R1, R2, Rx, Ry, R1 or R2 or O of this R control and then R negative blood and their remaining gene control Rh positive blood. So next we are going to learn Fisher's theory. Fisher's proposed that the three genes are involved in the protection of antigen and this three genes are lying so close together on the chromosomes that they act of one genes. Such genes are known as pseudo-alleles. He proposed a symbol of the three dominant gene as CDE. Are the recessive alleles a small c, small d, small e. A Rh negative persons should have a genotypic a small c, small d, or small e. If any one of the dominant gene is present, the persons will be Rh positive. Thank you.